How you feeling?
I'm ready.
Awesome.
Okay.
Welcome back to Marine Biology at Home. Today, I'm excited to tell you that we are being visited by Dr. Emily Brownlee, who is an expert in marine phytoplankton. Welcome.
Thank you so much for having me. I'm really excited to be here. I'm a classically trained oceanographer, biological oceanographer, but with an emphasis on phytoplankton ecology. So I love to talk about phytoplankton.
That is so good to hear because we're very excited to hear all about phytoplankton this week. Where can people find you and follow you?
Right. So I am an assistant biology professor at St. Mary's College of Maryland. And so you can find my email here, efbrownlee@smcm.edu, or very easily Google me, emilybrownlee.com.
Emily, why are you excited about phytoplankton?
Yeah, so I think what excites me most about the phytoplankton is the wide diversity that we find. So diversity in form or morphology, diversity in function, what they can do. And there's this perpetual mystery of why we see such diversity. It's called the paradox of the plankton. Why, when they're all competing for what is thought to be similar resources, do we see such diversity?
All right. Well, I look forward to learning this.
Okay, so phytoplankton, when we think of the word phytoplankton, it's really an assemblage of different taxa. Okay, so we have all different types of what we think of as single-celled organisms that have the ability to photosynthesize. You can maybe simply think of them as aquatic plants, but I don't like to put them into that category because they're single-celled and they have some really unique abilities, but also they're very diverse. When we think of these single-celled organisms that are photosynthesizing, we consider them plankton. Plankton are just organisms living in the water column. Plankton also means kind of tiny drifters.
Oh, that's a nice way of looking at it.
Yes, so and these guys are so small, they can't really determine their position very easily in the water column. So it's kind of like if we put ourselves into a pool of honey, we'd have a really hard time moving around. And that has to do because honey is very viscous for us. And because plankton are so small, the water is very viscous for them.
We talked about viscosity in previous lectures.
Great, I'm glad you did. And so they can't really counter typical ocean currents. And this is in contrast to maybe some of those charismatic megafauna you might think of, like a whale shark or fish that are able to determine where they want to be in the water column, and they can exercise movement against the water flow. If we think broadly of plankton, maybe not just phytoplankton, but all of those drifters, those tiny drifters, we can put them into categories by how they feed. And something like phytoplankton, which is going to be the majority of the talk today, are able to make their own food. Okay, they are photosynthetic. So they are able to use the sun's energy to convert carbon dioxide into an organic form of food. So they are kind of the base of the food chain.
Understood.
Yes. And so then we have those that are the zooplankton. So these would be considered the animals living as plankton. And these animals are feeding on the phytoplankton. So you can consider some of these as herbivores, or we think of them as heterotrophic. They feed on something else. And then we can have a mixture, all right? So we can have mixoplankton. These guys may function as animals, so they can feed, but they also sometimes can photosynthesize. And that's actually what I like to study. I study mixotrophic plankton that essentially act like aquatic Venus flytraps.
All right.
So they're able to both act like plants and animals. Plankton can also be defined as the amount of time they spend as plankton. This is going to be more applicable when you guys get to talk about zooplankton.
All right. So more of the animals.
Yes, so more of the animals. holoplankton are planktonic organisms that complete their entire life cycle as plankton. So those would be the phytoplankton. And then meroplankton are those that spend only parts of their life cycle as plankton. So a lot of times, benthic organisms like corals or sea anemones, their larval forms are planktonic, and this is what allows the sessile organism to spread and eventually spends the rest of its life rooted somewhere.
We'll actually have a lecture on larval ecology coming up in a couple.
Oh, wonderful. I love that. It's one of my favorites.
There's another way of differentiating plankton as well. There's how much exposure each group of plankton has. So most types of plankton are fully submerged into the water. But there are two additional types. There's what's called the neuston, which are plankton that are associated with the water surface. And frequently this could be things like a skim of bacteria on the very surface. So it's associated with the surface film. And then there's a few types that are regarded as pleuston, which are plankton that live at the surface but protrude into the air. And this is exemplified by Portuguese man-o-war and other siphonophores or jellyfish relatives that have a float or a sail that protrudes out of the water. So that's another way of dividing up the plankton.
And of course, another way to define groups of plankton are by size. And this really originally came from how we're able to sample plankton. So if you're using nets with a certain mesh size, the organisms that are smaller than the mesh size of the nets will remain in the nets, and you can collect them and look at them later. So this then we started to find that different size organisms were very associated with a certain group. So we have some of our smallest, the picoplankton. So these are 0.2 to 2 micrometers. The nanoplankton, so 2 to 20 micrometers. That's where we're going to find a lot of our phytoplankton in that size class, but they also can move into the microplankton size class, ranging up from 20 to 200 micrometers.
All right.
Yes.
I actually have a question for you. So those great big things up at the top, right? They've got jellyfish and sargassum, floating algae and other things up there. Are those really plankton to you?
Yes, they are. I think some of them are plankton in that they can't always determine where they want to be. They are drifting.
Okay.
So yes, we can consider them plankton. So as I said at the beginning, phytoplankton are really an assemblage of taxa. And we'll be talking about the main groups that you would find in the ocean. And they take a variety of forms, there's a variety of sizes, and also have a variety of impacts on both the oceanic ecosystems and humans as well. The first class that I want to talk about are the diatoms. So the diatoms are a really prevalent group. They're one of the most abundant that you'll find. Diatoms are made of this silica frustule. So you can think of it as almost like two pieces of glass, like a petri dish. So you can see here this centric or this circular diatom up in the top right, and think of that as, yes, it's like the petri dish. So two pieces of glass that are encasing this organism.
So like a little box.
Yes, exactly. And one of those valves is usually larger than the other one. Okay, so when you do take a petri dish, maybe the top is a little bit larger than the bottom.
Okay.
And you can see here in the top right that sometimes those single petri dishes will form a chain together.
All right, so you have a whole stack of little petri dishes that are composed of two parts, a top lid and a bottom lid, if you will, right?
Yes, and we call those the epitheca and the hypotheca. So these diatoms are fairly non-modal. They don't have flagella or these appendages that allow them to move. So these guys can be relatively fast sinking, but they are potentially able to decide or be able to determine where they want to be in the water column by creating vacuoles, which allow them to be a little bit more buoyant. So they're not just these fast sinking glass houses.
So these organisms will actually add a buoyant oil to the inside of the little glass boxes.
Yes.
To aid them in essentially floating upwards. Is that correct?
Yes.
All right.
And some of them, as you can see in this chain up on the top right, they have spines. So that might be kind of like if you were trying to swim and you didn't want to sink and you put your arms straight out to keep from sinking. Yes. Diatoms have a really interesting reproduction. So they usually undertake asexual fission. Okay, they have a fairly rapid doubling time of 1 to 2 times per day. And essentially what happens is that the two valves, the top valve, the epitheca, separates from the bottom valve, the hypotheca.
Okay, so we've taken apart our petri dish.
Yes.
Okay.
And the epitheca will establish, that's the larger size, that's that top of the petri dish, will then create another hypotheca. And eventually, and that goes on, and we can maintain some large size cells. But most of the petri dishes will start to get smaller and smaller due to the hypotheca.
Okay, so the smaller valve also becomes an epitheca, and so that reduces the overall size of the population.
Yes.
All right, well, that seems strange because you would always end up with a forever getting smaller collection of cells except for the few that are coming from the epitheca. Is that correct? How does that work?
So eventually they go through another part of their life cycle where they reestablish their size. And so they will create an oxospore.
There's a word for you.
Yes. Which eventually helps to bring them back to the large original size. Why are diatoms important? Well, diatoms are, for one, highly abundant in the oceans, like I've mentioned before. And they're very fast growers. They are quick to respond to increases of light and nutrients. So typically, they are responsible for the spring bloom that we get in the North Atlantic. They're also very important food sources for organisms like krill, which in turn are very important food sources for higher order or for larger organisms like cetaceans.
Okay, so basically this is the basis of the entire thing. We've got our little diatoms that are getting eaten by krill, which are then being eaten in huge number by the whales.
Exactly. Diatoms also, on the flip side, can be harmful to organisms.
How is that?
So they've been known to create domoic acid, or what we just call DA, and this causes a type of what we call amnesic shellfish poisoning.
Oh, that doesn't sound good.
No. So what it does is that it accumulates into filter feeders like shellfish, or even different sardines or anchovies that might have fed on the diatoms. And when larger organisms like sea lions, otters, ourselves eat those contaminated animals, this poisoning may result in us.
Okay, so that's why we've got dead pelican and 6 sea lion down on the bottom there, is because
yes.
the toxins from the diatoms have moved up through the food chain.
Right. And they may, when the toxins have moved to maybe the sardines or the shellfish, they may not be harmful to those, but they're harmful to us and to other types of marine mammals.
Oh, that's interesting.
Many times what you find is a fishery closure due to high amounts of these toxins found within the shellfish.
So people will close the clam beds or other things that are filtering the diatoms out and accumulating those toxins.
Yes, exactly. So the next major group of phytoplankton are what we consider the dinoflagellates. So dinoflagellates have this organic test we call theca or plates. They're highly characterized by their two flagella. So flagella are these appendages that allow them to move around. So dinoflagellates can, while they're still considered plankton, they do have a little bit of the ability to determine vertically where they want to be in the water column, whether they want to be near the surface or maybe they want to be a little bit deeper where they might have an influx of nutrients.
You're telling me that these are plants that swim?
Yes.
Very cool.
Yes. Many of these are considered heterotrophic. So I mentioned what that means. While they can photosynthesize, they can also feed. So they can be considered mixoplankton.
Oh, I know that this is your thing, and so I'm really excited to hear about it. But does that mean that dinoflagellates are essentially plants that can move and have mouths somehow?
They are able to ingest. So yes, they are able to engulf their prey.
Very cool.
Yes. And many times this is in response to a limiting factor that would limit their photosynthesis. So maybe in low light levels, they don't have enough sun to give them energy to photosynthesize. That's when they decide they want to feed.
Ah, that makes sense.
Yes. And in other cases, it can be maybe a limitation of nutrients. And these guys are quite abundant in the tropics and also in mid-latitudes in the summer. So right now I am close to the Chesapeake Bay. We get a large abundance of dinoflagellates here coming up this summer. So here's just another view of some of those dinoflagellates. You can see some of them have these armored plates on them, these theca plates made out of cellulose. They don't have that external silica skeleton like the diatoms do. And a lot of these reproduce by simple fission, so they don't have that reduction in size that you find in the diatoms.
All right, so no glass box. And by simple fission, you just mean that they break in half, right?
Yes.
Okay.
Yes, these guys will just break in half. And again, they're characterized by usually their two flagella, which lay in these kind of grooves, so that there's these grooves around the organisms you can see in some of these images called the sulcus and the cingulum. And these grooves are usually kind of homes for where the flagella can reside.
So it looks as though the sulcus is where the one little whip that creates forward motion would go, and the girdle or cingulum would be where horizontal movement goes?
Definitely. Exactly.
Okay.
So diatoms are not the only phytoplankton taxa that create toxins. Dinoflagellates are also major toxin formers, and they can create what we call, you might know or have heard of, red tides.
Yes. And look at that picture. I can see
Yes.
why they call it red tide.
Right. And so this has to do with the pigment that that's found within the phytoplankton. And when you have phytoplankton, even though they're microscopic and high enough numbers, then you can start to visualize this pigment. And it's not just red. There can be many different colors. It can be brown, mahogany. But when you start to see this color in the water, you know that it's probably a dinoflagellate bloom.
So when you say bloom, that just means an area of extreme density of these individual cells, correct?
Yes, exactly. You can get up to hundreds of thousands of cells per mil per very small volume of water.
Wow, big numbers.
Very big numbers for very small organisms. And so one of the major toxins is called paralytic shellfish poisoning. And these are a very potent neurotoxin, or we can call them brevetoxins, because they many times can come from one type of dinoflagellate called Carenia brevis. And this is just one example of the types of toxins you can find. These toxins, just like those in the diatoms, can accumulate in invertebrates that are then consumed by by things like fish or us. And this essentially, in lower levels, it can make you feel like you're drunk.
Really?
Yes. But in higher levels, it can lead to mortality. And there's also other types of toxins, like ciguatera toxins. This is another type of dinoflagellate that, when consumed, can make you sense things are hot. Things that are hot can feel cold. Things that are cold can feel hot.
This does not sound good either.
No.
These little things are bad news.
Yes, they are. And it's also not just the toxins, but when you have cells in such high number, it can block out light from organisms below it. It can clog fish gills. It also, when you have so many of these organisms, they eventually can only sustain themselves for so long. And when they reach the end of their bloom, they die. They can fall to the bottom, and there they start to get decomposed by bacteria. So bacteria start to break them down, and while bacteria do this, they use up the oxygen in the water. So effectively, these large blooms can cause oxygen dead zones.
So you're saying that the dead zones that we hear about are not from the living phytoplankton, but they're actually from the phytoplankton dying and then being consumed by bacteria, correct?
Right. Another group are the coccolithophores. These guys, again, unicellular, photosynthetic. They are really interesting, is that their kind of form of protection are these calcium carbonate plates, or what we call liths.
They're beautiful.
Aren't they? Yeah, they have all of these very intricate structures, and you can find them very widespread. They create high zones of primary productivity, so they're photosynthesizing a lot. And what's really cool about these guys is that because they have those calcium carbonate plates, that calcium carbonate is very reflective, or it has what we call high albedo. And so they reflect a lot of light and heat back to space, so that light and heat doesn't heat the oceans.
Oh, wow.
Right. And they also take in a lot of CO2. So photosynthesis fixes carbon dioxide, or CO2, and into an organic form. They take in CO2 not only from photosynthesizing, but also they need that carbon for their liths. Essentially what I'm trying to say is they help make the ocean a carbon sink.
You're saying that these phytoplankton are highly reflective, so they're having an impact potentially on global warming that way.
Yes.
That they're also hugely abundant, like we can see in your picture down there, that's coccolithophores from space, right?
Right, yes.
So they're hugely abundant, so they're taking in all of that calcium carbonate and all that carbon from the atmosphere, and that has to go somewhere. So when these things die, they're sinking down to the bottom, creating structures that we can think of like the cliffs of Dover, if you've ever heard of that, and taking tons of carbon out of the atmosphere.
Exactly. So those cliffs of Dover are essentially made of coccolithophores.
Wow. So tiny, tiny little organisms playing massive role in the way that the world works.
Yes, exactly. And some of the less abundant phytoplankton are things like silicoflagellates. These guys, similar to the diatoms, they do use silica, and they make these really intricate skeletons. You find these in more of the polar regions, so around Antarctica. There's some green algae that usually you'll find in estuaries, something like the Chesapeake Bay, and usually near the late summer and early fall. And then we have the cryptophytes, or the cryptomonads.
I don't know these guys at all.
So the cryptophytes are essentially, they're just small flagellated phytoplankton, and it's thought that they're not highly abundant in the oceans, but in coastal regions sometimes you can find very high abundances of cryptophytes. And it's been thought that cryptophytes are major food sources for some of the larger plankton, some of the larger, so the microzooplankton.
Okay,
yes.
So these are like the little bunnies.
Yes.
Okay.
Yes. We've just been talking about the eukaryotic domain of life. Okay. There's also some bacteria that can also photosynthesize. We call photosynthesizing bacteria cyanobacteria. And actually, some of this marine bacteria is some of the most abundant marine phytoplankton. Okay, so they can make up at least half of the world's ocean's photosynthetic biomass.
So that's kind of the elephant in the room here, is that we're talking about one group, but really there are these two other entire domains of life playing huge roles, and we're just kind of skipping them for right now.
Right, exactly. They're a whole nother lecture.
Emily, last time that we spoke as a class, I was trying to make the connection between the abiotic factors that drive life on this planet and how we actually take that into marine biology. And I think that with phytoplankton being the first step, maybe this is a place that you could explain a little bit more for us. So how do we connect light to primary productivity, or how much of that is actually coming from the plankton?
Right. So a lot of that productivity, almost 95% of it is coming from pelagic phytoplankton. And this all is originating from energy provided by the sun. So that light, that abiotic factor. Photosynthesizing organisms need light to drive that fixation of carbon into an organic form.
All right, can you break that down for us?
Yes.
What do you mean by fixation of carbon?
Right. Generally, the process of photosynthesis fixes or takes in carbon dioxide from the atmosphere.
Okay.
And this carbon dioxide can then get broken down into and fixed into an organic form called glucose. And when I say organic, this type of form can now be utilized as energy for higher trophic levels.
Okay, so we're essentially describing photosynthesis as the recipe of how do we cook carbon dioxide into food.
That is a great way to put it, yes. And there's some byproducts of that. So the process of photosynthesis will take water and split the water and create oxygen.
Oh, that's a benefit.
Very big benefit. So the phytoplankton in our oceans are creating over half the air we breathe.
Wow.
So the phytoplankton are creating over half of the oxygen in the air we breathe.
That's amazing.
Yeah. And so you can see here that sun is a major driving factor, or light is a major driving factor, and that photosynthesis is only possible when you have enough of this resource.
Right, and we talked about that a little bit in the last lecture, how that the light only extends to a certain point in water.
Right, so you have the uppermost layer is what we consider the photic zone.
Right.
And so many phytoplankton are limited. They're staying within that photic zone. They want to be able to get light.
That makes a lot of sense.
Yep. And so the light penetration, though, is based on a lot of different factors. So at the surface, we get a lot of light, but as light moves down towards the water column, things like turbidity in the water.
That's how stirred up it is, right?
Exactly. What also can affect it is the angle of sunlight. So we know that we get more sunlight in the summertime in the northern hemisphere, less in the wintertime in the northern hemisphere.
That makes sense.
The different wavelengths of light can also penetrate differently through a water column. So the radiation coming from the sun can range in wavelengths from about 290 to 3,000 nanometers.
So that's the total amount of radiation. So that includes the visible light that we see, correct?
Correct, because phytoplankton typically are using that visible light range.
Okay.
And so that's 400 to 700 nanometers. And that visible light range can penetrate differently throughout the water column.
All right.
So typically, the shorter wavelengths, so the blue wavelengths, will penetrate deeper into the water.
I think I see where you're going here.
Yes. And the shorter wavelengths are, like red, are absorbed higher in the water column. So that red color does not penetrate deep in the water column. And many times you can find some organisms in the deep that are actually red because you can't see that color.
And we can see that in this graph that you've got down here, where the deepest penetration of color occurs with the shortest wavelengths of light. So that blue color makes it down deepest, and the red colors are not getting nearly as deep.
And so that might explain why our oceans are blue, because that light is not, that color, that wavelength of light is not being absorbed. It's either transmitting or being reflected back.
I'll bet that's a question many people had until just now.
Yes. Outside of the visible light region, we can have wavelengths longer than 700 nanometers, and this would be the infrared.
Right.
And yeah,
how you keep your fries warm.
Exactly. And most of this is absorbed within that top bit, the first meter of the ocean. So you can see here on the graph that the absorption coefficient is much higher than some of those visible light wavelengths. And so this is what causes a lot of heating at the Earth or at the ocean surface.
All right, so let me get this straight. We've got our visible red light dropping out first of the colors, and then beyond the visible red light is actually infrared, which is heat.
Yes.
And that heat drops out even faster than the visible red.
Right.
So if there is solar heating of the ocean going on, it's only getting so far.
Exactly. You're mostly going to find that at the surface.
Very cool.
So absorption of light can be very dependent on the angle of which light strikes the surface of the water. If you're coming more at an angle of maybe 90 degrees, so straight down onto the water, then you can get more penetration of light. But if you're coming at an angle, then you might have a bit more reflection of that light.
All right. And so that's less light, right?
Right. Exactly. That angle at which light reaches the surface of water is called the angle of incidence. So that angle of incidence is directly related to the height of the sun above the horizon. So when you think about the tropics, the sun is directly overhead. This is at a 90-degree angle. And here is like having a flashlight shining directly onto maybe the wall in front of you. And it's a nice concentrated beam of light.
All right. So that, with what you just said, means that we've got the most light coming in at the tropics, which makes sense with what we've thought about so far. And it would also mean more heat at the tropics, which makes sense with what you've said so far.
Right. But if you start to move up in latitude, so maybe we get closer to the North Pole or even the South Pole, that angle of incidence becomes a bit smaller.
Okay.
And what that does is if you took a flashlight and maybe you shined it at an angle onto a surface, that light would be kind of more spread out. And so that light, for one, would not be as concentrated on the area. And also, when you have that smaller angle, then you're going to have more reflection of that light back. So you have less heating.
Which would mean loss. All right. I understand.
Yes. So in places like the Arctic and Antarctic, because of the seasonal differences, during the winter, the sun can be almost absent. And during the summer, we can have light almost all day.
Yeah, no, I think that makes a lot of sense, especially because it means light availability for the organisms that you study is going to vary tremendously based on where you are on the planet in the season.
Exactly. And so many regions, like maybe temperate regions, like the region that I live in, so here in Maryland, have very seasonal changes in primary productivity. So in the early spring, when we now are getting light after kind of our darker winter, we start to get blooms of phytoplankton, and they continue into the summer where we have maximum light. So we know that light is a major driving factor of primary productivity. And so when you have really low light levels, this can limit photosynthetic rates for species. And alternatively, if you have too much light, this can cause something called photoinhibition. And so when you look at this graph below, what we have is the amount of light on the x-axis and the relative photosynthesis on the y-axis. And interestingly enough, you can see that different taxa within that phytoplankton assemblage can or have adapted to photosynthesize at various light levels. In this case here, green algae reach their maximum relative photosynthetic rate at low light levels. So they've adapted to be able to deal with low light. Diatoms, in turn, can deal with kind of a mid-light intensity, while dinoflagellates are better adapted to higher amounts of light.
And you told us earlier that your dinoflagellates come in the summer, right? So this makes sense.
Exactly. They are able to compete better than the other organisms because they can deal with this high amount of light.
All right, that's interesting.
And so what this does is it shows that we can have different groups coexisting if they're inhabiting different niches. And so what we might have is a scenario like you see here, where in different in different depths of the water column, we can find different assemblages. So maybe the dinoflagellates are found closer to the surface of the water, while those that are better for low light levels, the chlorophytes or the green algae, you'd find deeper in the water column.
That makes a lot of sense to me based on what we've learned about phytoplankton from this lecture. But can you tell me, does it seem to occur to you in your system? Like, is there any truth to this?
So yeah, actually I have seen this in the system where going from the winter into the spring, we get a diatom bloom, but that diatom bloom is actually occurring a little bit further down in the water column. And more and closer to the surface, we find assemblage of dinoflagellates and also things like cryptophytes. So yes, we see this kind of depth partitioning of the organisms.
Very cool.
And we also see it seasonally as well. So those that are adapted for lower light levels are found mostly in the winter, while diatoms, which are responsible for the major spring bloom, which are adapted at or are well adapted at mid-light levels. And like you said earlier, the dinoflagellates occur in the summer, so they like that high light.
So that makes sense with what we've learned about the light availability at different places on the planet and at different times of year. How else can phytoplankton vary?
So phytoplankton can adapt not only to different light levels, but also for the other resource they need, which are nutrients. So phytoplankton need a little bit of extra food or inorganic nutrients that allow them to photosynthesize.
What falls under an inorganic nutrient? What are the types of things that do that?
So phosphates, and nitrate, nitrite can be considered inorganic nutrients. Silica can be found, so those that are needed for the glass houses that the diatoms live in. All of these are essential for their growth and their photosynthetic activity.
All right, so you're saying that light is an important limitation, but there are other things that limit the growth or can aid in the growth of phytoplankton communities.
Exactly. And nutrients can be variable by location, by time of year. And I think that's a whole, maybe another lecture to get into. But essentially there are taxa that have adapted to different amounts of nutrients. And so what I have here is just an example of marine cyanobacteria. So that bacteria, that prokaryote that can photosynthesize. This is a profile of a water column. So the y-axis, as you're going down the y-axis, that's depth. Okay? And the x-axis is kind of the amount of the limiting factor, so light or nutrients. And what you can see here, light diminishes as you go further down into the water column.
So that's that orange dashed line.
Exactly. And that's where you have a lot of photosynthesis. So there's typically associated with that a drawdown of nutrients because those photosynthesizing organisms are using up the nutrients.
Ah, okay.
And as you go deeper, where there's typically less phytoplankton because there's less light, that means you have an increase of nutrients because they're using it up a little less. And what you find within a water column are different groups of this prochlorococcus that are adapted to high light levels or low nutrient levels, or those that are deeper that are adapted to low light levels and high nutrient levels.
Emily, if I understand what you're saying correctly, we are looking at some of the diversity that you were talking about in the very beginning. Does this graph help us explain your paradox of the plankton?
I think that this graph does. I think it shows that this diversity can coexist in the oceans because the oceans are not just in kind of a soup. There are minute variations in light. As you go down deeper into the water column, there's variations in different types of nutrients. And what these organisms have done is they've adapted to compete for these different types and these different variations of nutrients. And so what we see is a very wide diversity, even just within one organism.
So many more spots. We've got lots of different places within the oceans for different specializations to be important.
Exactly.
Thank you so much for spending your time with us. I really appreciate it.
Of course. Thank you for having me.
So next up is zooplankton. And we will have another guest lecturer. And then I also want to remind you about that you can ask questions at the Facebook group, Marine Biology at Home. And we have a number of great people supporting this work behind the scenes. Also, please get in touch. If you are in a position to offer a video or a explainer to any of our lessons, if you would like to donate your time in video editing or sound editing, all of that would be greatly appreciated. So please reach out if you're finding what we're doing valuable. Thanks.